and the environmental are still unknown. So, in this communication, we have explored the possibilities of single particle ICP-MS. What is ICP-MS? This is inductively coupled plasma mass spectroscopy for the quantum and the characterizing titanium dioxide and silver nanoparticles in human urine. Inductively coupled plasma mass spectrometry operating as single particle mode has emerged as an efficient vertical technique for quantific and the characterizing this is size distribution of the metallic nanoparticles. But the development methods have been validated and applied to several human urine samples. But what about the ICP-MS? ICP-MS, as I told, this is inductively coupled plasma mass spectrometry, or the ICP-MS, is an analytical technique used for the elemental determinations. So employs a plasma as ionization source and a mass spectrometer analyzer for detect ion. Also, it can perform qualitative, semi-qualitative, and the quantitative analysis. And this method developed in 1980. Yeah. So this is our ICP-MS. Using this instrument, I performed my work. Which methods for the nanoparticles characterization we have? This is single, as I told, single particle ICP-MS. But what is single particle ICP-MS? Each nanoparticle gives a transient signal, a plume of ions generated from the particle, but use time resolved data collisions and the analysis. Particle concentration, particle diameter, composition, and size distribution are obtained. You can see here we have nanoparticle sample, and we can detect. So after detection, we will see on ecran of the computer signal from the nanoparticle event, where we have the dependence of signal intensity from the time. What is ICP event? How can we analyze our compounds which contain nanoparticles? You know that packaging. In our packaging, we have a lot of nanoparticles. But in this packaging, we keep our foods. From our foods, we can take these foods. But from these foods in our organism will keep a lot of nanoparticles. They can be toxic or not toxic, but we must characterize these nanoparticles. But how? Using ICP-MS. Using ICP-MS. Single particle ICP-MS. For the nanoparticle, every time we will use a single particle ICP-MS. But ICP-MS we will use just for the total characterization of the metals. But in our work, we used urine. This is sample pre-treatment. We can have voice. Okay, thank you very much. 1 milliliter of urine. 1 milliliter of urine, we diluted up to 10 milliliter with 1% solution of nitric acid. So we used ICP-MS, where during the measure with the ICP-MS, we use standards, internal standards. They are ruthenium, scandium, and germanium. But it's in the coin duplicate samples analyzed in the triplicate. But titanium dioxide nanoparticles, also by this way, we analyzed silver nanoparticles. 1 milliliter, this is single for the single particle ICP-MS, 1 milliliter of urine. We diluted up to 10 milliliter with 1% solution of the glycerol. So titanium dioxide nanoparticles density during measure with ICP-MS, we had titanium nanoparticle density 4.23, mass fraction 59.9, sample flow 0.41, 0.45 milliliter minute minus 1. Transport efficiency 2.3, 5.6, and dwell time 50. Mass ratio 48.948. So in this case also we used ultrasound steering just before measurement. Okay, before measurement every time we use ultrasound steering. So all urine samples used in the current research were from healthy volunteer adults staffed at the University of Santiago de Compostela. Sample collection was performed in accordance with the ethical committee for the clinical research of Galicia. Galicia is part of Spain. Then which gets a written informed consent from all volunteers. But urine samples we took 10 milliliter, were collected in pre-cleaned polypropylene wells and were stored at 4 degrees of Celsius. This is for a maximum of 48 hours before analysis. But total silver and titanium determinations were performed with the Nexon 300. This is a mark of the ICP-MS, this Perkin-Elmer. Welcome, may you say, equipment with the nebulization system composed of a glass concentric nebulizer. The glass cyclonic spray chamber thermostat by a Peltier refrigerator, then see first outer sample, this elemental scientific OMAPA MBA USA. The same ICP-MS instrument has been used for the silver nanoparticles and titanium dioxide nanoparticles assessment. Conjugation and size distribution by using the nano application software version, Perkin-Elmer. Other devices were Piper 150 ultrasonic water bath from Espinar, this is Barcelona, Spain, and the Labo Centrifugan 2K15. This is centrifuge from the Sigma. This is from the Germany. But before measure, we centrifuge urine. Why we need in centrifugation? We must you know that in urine we have some amount of the protein, because we must differ this protein from urine, and for this we must use centrifugation. But usually in the centrifugated urine we found not a lot of nanoparticles, but in not centrifugated urine we found more amount nanoparticles than in the centrifugated. So in this table, you can see ICP-MS and single particle ICP-MS instrumental parameters for the total silver and titanium determination, and 4 nanoparticles containing silver titanium dioxide determination characterization. So, for example, here we have the radio for the frequency power, gas flows. Then we have the Keter mode, where for the analytes we used the internal standards, as I told, the ruthenium and the germanium. Then we analyze, this is analyze the silver and the titanium. Operating single particle ICP-MS conditions, they are nanoparticles containing silver, then a mass of the silver, density, mass fraction, ionization efficiency, sample flow rate, dwell time, sampling time, mode, number of the readings, then replicates, nanoparticles which contain titanium dioxide, mass of the titanium, density, mass fraction, ionization efficiency, and other parameters for this measure. For the nanoparticles containing titanium dioxide size distribution in some human urine. Mean size for the nanoparticles which contain titanium dioxide varied from 76 to 98 nanometer. Most fragrant sizes from 70 to 68 nanometer, 86 nanometer. A similarity between the mean and most fragrant size shows that nanoparticles containing titanium dioxide size distribution follow a log normal distribution. So in this slide, you can see silver nanoparticle size distribution from silver nanoparticles, where we use the size 16 nanometer. Standards and human urine samples picked with the silver nanoparticles with same size 16 nanometer. Okay, this is for the silver, just for silver nanoparticles, and in this case you can see urine and plus silver nanoparticles which have the size 60 nanometer. This is nanoparticles which containing silver size distribution in some human urine. There are nanoparticles which contain titanium dioxide size distribution in some human urine. For example, for the urine sample 2, 3, 4, and for the urine sample 10. Because we took 12 volunteers for the urine sample. So after each last we used lot and log for the calculation. We calculated lot and log. And from slide, you can see these calculations. Dear guests, can you hear me? Dear guests, can you hear me?
Yes, we can hear you, but there is no presentation.
Okay. Can you see now presentation?
No, we can't.
Now, can you see?
Now it's visible. Okay, it's fine now.
Okay. Before I thought that we used the concentrations for the nanoparticles, and we calculated load and log. And from the slide, you can see these calculations where we used also blank value method, dissolved calibration method. And also we used repeatability there where we have nanoparticles concentration, nanoparticle size, most frequent size here for the titanium dioxide and for the silver, and the analytical recovery. We calculated these parameters for the 12 urine samples. But analytical recovery we calculated for the 9 urine samples. So, application to human urine samples, as I told, we took 12 urine samples, and where we have our samples, then total titanium dioxide nanoparticles, their concentration of them, of these particles, and the titanium dioxide nanoparticle size in nanometer. Also, we used other instrument. This is zeta sizer nano ZS90. Affordable molecular particle size and zeta potential analyzer. An entry-level system for the measurement of particle size and a molecular size at 90 degrees scattering angle using dynamic light scattering, also with the ability to measure zeta potential and electrophotic mobility using a laser Doppler. Microelectrophoresis and molecular weight using static light scattering. They are our team in Santiago de Compostela University, where I performed this work. This is size distribution by the intensity, where we have the dependence on intensity on the size. So, imaging what we use for this work. Imaging-based techniques, they are scanning electron microscopy, SEM, and transmission electron microscopy. We performed this work at the center of the science and the technology which we had at the Santiago de Compostela University. And this is our work. We perform the performance of this work to the term of the SEM. Okay, the SEM scanning electron microscope image the woman 37H urine, then a temp image of healthy adults men 48H. And then also we have a same image of adults, women 25H. I thought that we took the urine sample from the different persons, from different persons, healthy person and persons. And in conclusion of this work, I can say that single particle ICP-MS has been found to be an applying analytical technique for assessing metallic nanoparticles in complex sample, such as urine. The developed method were able to detect and quantify titanium dioxide nanoparticles at low concentration, and also small nanoparticle size can be assessed. But human urine sample can be directly analyzed after deletion 1 to 10 with 1% solution of the glycerol. But centrifugation must be omitted because nanoparticles are partially lost. The methods are repeatable and good analytical recovery. So several nanoparticles concentration and nanoparticle size were observed. Titanium dioxide nanoparticle size within the 4100 nanometer range were quantified in most human urine samples. Which knowledge we have here. This work was supported by the Spanish government. This is our project. It was under the Erasmus Mundus Media framework. So for it, I am very grateful to the education, audiovisual and cultural executive agency, European Commission for an Erasmus Mundus Action research, media research grant. It was our project, and as a result of this work, our article, our paper, possibilities of single particle ICP-MS was determining, characterizing titanium dioxide and silver nanoparticles in human urine, was published at the International Journal of Trace Elements in Medicine and Biology. This is so.
Thank you so much. Please carry on. Carry on.
So sorry.
I guess you have finished. So I just thank you. I think 2 slides are remaining as well. You can continue, please. I'm sorry, ma'am, for the interruption.
Sorry, at last, in end of my presentation, I will describe my country. You know that my country is Azerbaijan, where we have the government form republic, where Baku is capital of Azerbaijan, population more than 10 million, but area you can see 86.600 kilometers squares. So, is our national dance, our national instruments, the monuments, historical monuments. So, as our the monument by name is Kızgalası, then this mosque, inner city. You know that Baku has inner city and the modern city. This is our modern city. They are inner city. This is our Azerbaijan, my Azerbaijan Medical University, where I work. We have several buildings. They are my students from different Muslim countries, from Iran, from Iraq, from Algeria, from Palestine, from Syria, from Sudan, and others. They are my workplace in Spain at Santiago de Compostela University with ICP-MS, and they are Nobel laureates. The Nobel laureates. Then this is my team, Getter, by name Getter. They are a team from the Santiago de Compostela University from Spain. First, thank you very much for attention, dear guests. Thank you very much.
Thank you so much, ma'am Kamala, for a wonderful and informative session from your side. There are some disturbance during the presentation, and I'm very sorry for that. But they are also a part of our session, so there is a bit hard for me to arrange all these things at once. So thank you so much. And at the end of this session, we have some questions for you in the chat box. I will ask you one by one, which I see in the chat box. Okay, before that, I want to say that we as a Pakistani, we support Azerbaijan, and we really respect the people of Azerbaijan.
Thank you very much. Thank you very
Okay,
much.
there is a question from Sangeeta. She is asking that what is the concentration used for metal nanoparticles in the in vitro assays?
Sorry, can you repeat?
Sangeeta is asking what is the concentration used for metal nanoparticles in the in vitro assays?
In vitro.
Yes.
In vitro. Okay, which nanoparticles? Yes.
What is the concentration
What is
of the
concentration? What is
nanoparticle?
concentration used for metal nanoparticles in the in vitro assays? Okay,
Yes.
what about the concentration? As I told, we use just 10 milliliters. 10 milliliters for in vitro. We used not in vivo. We used in vitro. We didn't inject, for example, the humans with nanoparticles. We used just in vitro assays, and here we used 10 milliliters of each for each nanoparticles. And at last, for example, here we have some concentration which we used for this assay. For example, now I will use a concentration of the titanium dioxide nanoparticles was here. I thought that about 10 milliliter, and usually we used 1 milliliter.
Okay. I think she has got her answer. Now move to the another question, which is from Omar Liaquat. Okay, he is asking, what is the use of nanoparticles against insects? Have you got the question?
Let's see.
Can you hear me? Hello,
What
Miss
is the
Kamala?
use? What is the use of nanoparticles
Nanoparticles,
against
insects.
insects? Yes. What is the use of nanoparticles against insects? It means which nanoparticles or what?
I think he is asking generalized nanoparticles.
Generalized nanoparticles. Usually for insects, I didn't hear about these nanoparticles which we can use against insects. For example, we have just nanoparticles. Maybe in literature we have about it, but in my case, we didn't use these nanoparticles, and we have just nanoparticles which we can use in medicine, for example, for cancer, for other diseases. Just use the small size of these particles.
Okay, we have another question from Bahadin Ahmed. He is asking that could we doing nanoparticle test for someone who hold cancer through blood sample?
Okay, could we doing nanoparticle test for someone who hold cancer through blood sample? Yes, yes, we can. We can doing this nanoparticle test. Why? Because we wanted to continue this research, but we hadn't a lot of time for it. Just we stopped in urine sample, but through blood sample, yes, we can. Other question?
Okay, someone is asking, can you share the slides?
What?
Can you share the slides? which
Can
you
I
have
share?
presented today.
Can I share this slide?
Uh,
Which slide?
the whole presentation.
Can you see?
He is asking about the whole file, PPT file, PowerPoint presentation file, the whole PowerPoint presentation file.
As a gate in can we use to treat? This is for the question of Farhana Bano.
Okay, there is another question from Hurulan. How long do nanoparticles stay in the body?
That I will see.
It is some much.
If you use nanoparticles, this one or not? Which nanoparticle is used as a treatment of disease, or which question?
I think she is asking about the treatment, I guess. Okay. How long do nanoparticles stay in the body? When you use the nanoparticles to treat or diagnose something, how much the time they stay in the body?
It depends of the kind of nanoparticles, which nanoparticles. This is, for example, usually we have the nanocapsules, we have metal nanoparticles. It depends with what kind of the nanoparticle.
Okay, we have.
And also it depends on total organism of human.
Okay. Okay, we have another question from Farhana Bano. She is asking if we use the nano drugs, then are any serious effect to human body?
Usually they haven't serious side effect to human body, but nanodrugs they have very good, but usually they have very good effect to our body in several diseases. But serious effect, it means maybe a toxic effect. Serious effect if we will use more toxic or the very in the very large dose.
Okay. There, Rao Zubair is want to ask. Rao Zubair, just write the question in the chat box if you want to ask. I can't take any call from you because we have a lot of people. Okay, we have a question from Sabha server. Which nanoparticle is used in the treatment of typhoid?
Which nanoparticles?
Used for the treatment of typhoid.
Usually gold nanoparticles. Usually gold nanoparticles is very used. Silver nanoparticles are very used medicinal drug.
Okay, we have a question from Muhammad Usman. He is asking, what was the impact of your prepared nanoparticles on immune system?
Immune system. But now we have the problem with corona, yes? But usually nano drugs, they can increase immune system in our organism, but also it depends on the nanoparticles, it depends on the drugs which we use, but usually they increase. But maybe some organism can affect different.
Okay. We have question from Adil Akram. He is asking, which nanoparticle are more effective, natural or synthetic? as he has isolated zinc oxide nanoparticles from Azari Azadi Rata indica.
Mhm.
It shows antibacterial activity against resistant isolates of E. coli and Klebsiella species. Sorry for the pronunciation. I didn't know about it.
Okay.
Uh,
Which nanoparticles, artificial or synthetic or the natural nanoparticles, they are very effective. Yes, I think. First one. Maybe each student, each scientist knows that usually very effective, maybe not natural, but natural nanoparticles or the natural drugs. Natural drugs, they are plant origin, usually plant origin, or but they will affect to our organism not fastly, but chemical synthetic nanoparticles, they will affect to our organism very fastly, because but effectivity of the natural drugs or the natural nanoparticle drugs, they will be very good effectivity.
okay, Fatma Hussain is asking, which type of nanoparticle give optimal activity with fewer side effects for medicinal plants based nanomedicines?
Which side effects? Sorry.
Which type of nanoparticle give optimal activity with fewer side effects from medicinal plants based nanomedicines? If there is
From the medicinal.
okay, if there is a medicinal plant based nanomedicine, which type of nanoparticle have optimal activity with fewer side effects?
Um, Plant nanoparticles, they have very good activity every time, but medicinal drugs, usually they have side effects.
Okay, Muhammad Sarfraz Ali is asking, what is the role of nanoparticles in personalized drug? In personalized drug, what is the role of nanoparticles?
In
Personalized drugs, what is the role of nanoparticles?
okay, what is the side effect of nanoparticles in case that I will. So I want to find this question. So we have here one question. I don't know it was this question or not. Nanoparticles have industrial application or not? I don't know. This question was or not?
No, it is in the pipeline. I'm asking one by one.
Okay, now that I will find, because if I will find this question, if we use the nanodrugs, then is there any serious effect to human body? Then what is the relation?
Okay,
Relation.
we
So
have. I got the question
repeat,
from Khurram Shahzad.
repeat, please, repeat.
Okay, is there any consideration of nanomedicine to cope viral or infectious disease such as COVID-19? Is there any consideration of nanoparticles, nanomedicine? Is there any possibility of nanomedicine to cope the viral infection like COVID-19?
COVID-19, but I thought that we have the protein corona, yes? You know that COVID-19, what is COVID-19 virus? This is protein. This is protein which we can close, for example, this protein with nanoparticles too. With nanoparticles too, you know that this protein we can take from the plant. Yes, I think that some nanoparticles can affect to this virus too, in case of COVID-19.
In continuation, there is another question. Can silver nanoparticles be treated as potential drug for SARS-CoV-2
Yes,
coronavirus?
yes, yes. If some scientists will find this drug.
Okay. Abdul Malik is asking, how can we use iron-coated nanoparticles in molecular level?
Iron or which nanoparticles?
Iron-coated nanoparticles.
Iron.
Iron, Fe, ferrous, iron.
Iron nanoparticles, but usually I didn't use iron nanoparticles. I think that in molecular level we can use molecular iron nanoparticles. Why? Because you know that iron is most important composition of our organism, but in which part in the blood contain iron. I think, yes, iron we can use in molecular level.
Okay, Vakar is asking, by which technique we have determined the size of nanoparticle? How can we determine the size of nanoparticles?
By single particle, using a single particle ICP-MS, we have a lot of methods for the characterization, but a very good method. This is single particle ICP-MS.
Okay. Okay, does nanotechnology change our DNA? Is there the possibility? It can mutate our DNA, nanoparticles. What do you think?
Sorry, as
What do
this
you
as a,
it is a question from Malik Mohammed. He is
uh-huh.
asking, does nanotechnology change our DNA?
Nanotechnology is,
Can change our DNA?
of course, nanotechnology can change everything.
There is another question from Sabah.
Mhm.
How can I know the concentration of nanoparticle for a treatment of any disease?
For treatment of the nanoparticles, just in many literatures, we have this information. This information is about the treatment of some disease, but which concentration? Which concentration? It depends on which nanoparticles you will use for treatment and which disease. It depends.
Okay.
And usually it depends on the organism, on human organism.
Means it depends, the concentration of nanoparticle depends on the organism, how much it takes and how much it stabilize in the body. That's I got the answer. Okay, we have another question from Pankaj Hada. He is asking, how does nanoparticles affect the cell membrane?
Sorry, Dia,
How
can you repeat?
how does nanoparticles affect the cell membrane?
Membrane, cell membrane.
Ye- Yes, cell membrane. I
Yes,
think
of
cell
course.
membrane of bacteria, I guess.
Yes, of course, of course. And in presentation, which I shared before, I told that nanoparticles, toxicity of nanoparticles, can be showed in cell membrane, in a molecular level, and in other parts of the organism. Yes, of course.
Okay, Shahrukh Awan is asking, is there
Sorry,
any role?
sorry. Why cell membrane? You know that diameter of cell membrane is not large, and our nanoparticles, they have very a small diameter because they can include to our organism very easy and can affect to cell membrane too.
Okay, Shahrukh Awan is asking, is there any role of nanoparticles in early detection or diagnosis or prediction of any specific disease? Can you give any example of nanoparticle
Mhm.
which is used for early detection or prediction of any specific infection or disease? Ca- Have you got my question?
As I understand that you to ask me about nanoparticles, about artificial nanoparticles, yes or not?
No, no. I'm asking, uh, is there any role of nanoparticles
Role.
in role of nanoparticles
Uh-huh.
in early detection of any disease, early diagnosis of any disease?
Yes, of course. Of course. They have a very good role in detection and why just due a small size they can enter to each cell and they can detect. Yes, they have a very good role in detection.
Uh, he is also asking the example of that nanoparticle. Can you give the example? Which nanoparticle is used for early detection?
But for example, we can use also nanocapsules. Nanocapsules or the carbon mm, nanoparticles. Usually carbon nanoparticles, they are useful for this mm, poison, for this one.
Okay, we have another question. Nadeem Altaf. He is asking, can gold nanoparticles conjugated with natural peptides can be used as effective antiviral?
Okay, about the conjugation of the nanoparticles with natural, yes? And
Uh,
can they affect? Yes, of course.
is this effective against anti a virus?
Yes, they can affect, they can conjugate with natural compounds and also can affect, yes, of course. You know that in our organism also we have a lot of natural organic compounds. For example, they are amino acids, they are proteins, they are carbohydrates, but usually they can conjugate with proteins. And by this way can affect our organism. But maybe you know that I will say it depends on the country, depends on the area where you live. But for example, do you now I have question for you. Do you like to drink a milk?
Sorry,
Do you like to drink? Do you like milk? Milk.
milk.
Yes. But
Yes.
which milk do you like? Artificial or natural milk?
Natural milk from cow or
Oh,
buffalo
very good,
or
very good.
goats.
Why? Because artificial milk, which we have in packet, for example, which we can buy in the market or the supermarket, they contain a lot of nanoparticles. For example, titanium dioxide nanoparticles. Or do you use sun creams?
Yes.
Okay.
Milk.
They also do you use deodorants? Do you use perfumes? Also,
Yes.
they contain a lot of titanium dioxide nanoparticles. So, also, if they will enter to our organism, you know that in our organism is natural organism. They will affect to our organism, and from with each we can take a toxic effect. Yes, of course. But for example, a lot of seafoods also they contain nanoparticles. Not just nanoparticles, also they contain not all, but some seafoods they contain nanoparticles and also they contain heavy metals, which will affect our organism very toxic.
Okay, we have some raised hands as well from Abid Rahim.
Mhm.
Abid Rahim, I want to. Why don't you unmute? Wait a minute. Okay. I don't know why he's not unmute. Unmute yourself. Abid Rahim, you can unmute yourself. Khurram Shahzad, I have asked your question, I guess. Uh, then make an attempt the spotlight when not. Okay, we go to the conversation because there is a problem in.
Mhm.
What Khurram Shahzad is asking again, what is the ultimatum goal of nanoparticle scientist? Because there is an extensive research, many researchers are working on nanoparticles. So what is the ultimatum goal for researching more and more extensively in the nanoparticles?
It means in science or
Have you got my question? Or
for the in science or for environment in totally.
okay, you are saying that it is useful for the environment or any mankind benefit.
Elemental medicine. Yes, they have a big role for the environmental and for the medicine. Because I told in my presentation, I showed you that from the nanoparticles we have the biological toxicity and the environmental toxicity. For example, we can coach, we can work in industry. If we will work in industry, we can take a lot of toxic nanoparticles. It will be for us environmental nanotoxicity. But from the biological, you know that it will caused from the natural, from plants or from animals.
Okay, we have another question from Yasinullah. He is
Mashallah,
asked,
today a lot of questions.
yeah, a lot of questions. I choose some little question because when there is a long question, it is very hard for me to understand first and then try to ask you. So there is a little question. What is the relationship between nanochemistry and smart material?
Relation of the nanochemistry and the smart material, yes?
Yes.
Yes, every time we have a relation of, for example, two types of the compound, because in this case also we have a relation of both compounds. So, for example, I know that smart materials, they are also, for example, intelligent or the responsive materials, because in this case, we have some influence of the nanoparticles to the smart materials.
Okay. I have to clarify, Humaira Jahan, that nanoparticles are not dangerous until and unless it exceeds from the required concentration. So they are not so much dangerous, but are beneficial when we take in the required quantity, or if they are stable in position in our body. They are very helpful for the diagnosis and for the treatment of diseases. Okay,
Yes,
so they
yes,
are
they are
not
very helpful.
dangerous.
Yes, they are very helpful in diagnosis. They are dangerous, but in a very high dose, very high concentration, not in
Uh,
small concentration. But every drug, every medicine, or every chemical, you know that they are very toxic in quite concentration.
yeah.
Quite concentration, not in small.
Okay. Actually, I have also worked by synthesizing nanoparticles, nanoparticles, by using fungal species, and we have identified that they are producing nanoparticles, silver nanoparticles, in the presence of stress. We have given them stress, so they produce silver nanoparticles. Then we conjugate with the bacteriocin, an antibacterial compound, which also
Yes.
shows a lot of antimicrobial zones against the infectious pathogens.
But for example, silver.
So,
For example, silver
yes,
nanoparticles, they have antibacterial effect.
yes, and when we conjugate it with the bacteriocin, it also shows a very high susceptibility of pathogens against them. So, therefore, I have some knowledge of nanoparticles in this regard. Okay. We have question from Rausware again, that nanoparticles are photocatalysts. They
Okay.
produce they produce free radicals,
Mhm.
which are very toxic and cause mutation in DNA. How would we tackle this problem?
Okay, which one is very toxic? For example, nanoparticles, can we use as photocatalyst or not? Yes,
Uh,
maybe it was this question, yes? As photocatalyst. Can we use nanoparticles as photocatalyst, or which one is very toxic? Yes?
uh, he is asking, some nanoparticles are photocatalysts. They produce
Photocatalyst.
free radicals. They produce free radicals.
Mm-hmm.
So, which causes mutation in DNA. So,
Uh,
how can we tackle this problem?
Free radicals. You know that free radicals, they are not stable particles in chemistry because they can enter to each part of our organic compounds and they can influence.
Can I ask you more questions? Because there is a list of questions in the chat. Or we can. Okay, may I ask you more questions?
If you want, I will find the questions from here. For example, is nanoparticles is toxic or not? Of course, nanoparticles, some nanoparticles, they are toxic. As I told, in large dose, they are toxic. Then, for example, from Vagar, we have nanoparticles. How is the effect of the brain? Which disease should cause from nanoparticles? Of course, for example, titanium dioxide nanoparticles can affect to the central nervous system. To central nervous system, as I for example, I showed you before in the presentation that which disease we can take from the brain. They are neurological disease, they are Parkinson disease, and the Alzheimer disease. They are from the nanoparticles, but usually titanium dioxide nanoparticles, they are influence effect to central nervous system. Okay, other question that I will read, you will rest. Yes?
Okay, I have a request to you. Can you please show the first slide of your presentation?
Can I what?
Uh,
Share?
can 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 you show the first slide of your presentation?
Post like.
Yeah, first slide. The main title slide. We
Yes.
have to take a picture of it.
Yes.
Muzammal, please take the picture. Your presentation is not moving.
Okay, that I will show. Can you see?
Yes, I can see, but move it to the first slide. Can you? Okay. Thank you so much. We have to ta- take the picture. Okay, Muzammal, take the picture of the first slide you are asking about. Okay. Our organizer, another organizer is asking me to share the first slide for taking the picture. Okay. I think we have to sum up the today's session. Thank you, everyone. I hope you enjoyed a lot this informative session. I have already shared the feedback form link in the chat box. You can see and you can find it in the chat box. Another important announcement is that we will send you e-certificate for both registered participants who have attended the session and who have not registered. They can receive the e-certificate by filling the feedback form. Okay, so you have
Okay.
the chance.
Dear
Okay.
Firoz, can I ask, sorry, very sorry, but from my university also we have a lot of participants, maybe in your YouTube canal, so in YouTube channel. If they know, if they can see us by YouTube channel, how can they take this e-certificate? But usually e-certificate is very important than the speaker, maybe you know.
Okay, they will get the e-certificate by watching the YouTube recorded video of your session, and they have also the chance to get the e-certificate by filling the feedback form. We will share the link in the link of YouTube channel, so they can also get the certificate. Don't worry. Okay, we
Okay.
have
Mhm.
we have given a chance to our participant to get the e-certificate, so there is no problem. Or if someone uh, l- left for the e-certificate, please remind us. We will send certificate for the participant. Okay, from your university. If
Mhm.
some student left for the e-certificate. Okay,
Mhm.
but we are trying our best to sending the e-certificate to all our participants who attended and who have watched the streaming of yours our session in the YouTube channel. So there is an open chance for all the participants. So thank you so much, Kamala, um,
Thank you so much
for your information.
for your opportunity, for your organization of this very interesting event with very interesting questions. Thank you very much.
Thank you so much. Thank you so much, all the participants, for their patience and for your attention. I hope you have got your answer from the Kamala, and we are very thankful to you for your engagement.
Okay, I thank
Thank you
you for
so much, and thank you so much. Good good night, Kamala.
good night,
Okay,
good night, good night, dear.
good night.
Night.
So
Thank
this
you
is
again.
signing off, Dr. Rouge. Allah Hafiz.
Have a nice day, inshallah.
Inshallah.
Inshallah. Goodbye.
Allah Hafiz. Goodbye.